Bhavya is a PhD student in the Department of Biological Science. Yeah. I'm sorry. Okay, I'll go ahead. She's a PhD student at the Department of Biological Sciences at TIFR. She's working in some aspect of cell biology and neurobiology with a focus on understanding of regulators of membrane fusion. I also don't know about this regulators of membrane fusion because I'm from physics. But today she is going to talk about how biologists at TIFR study a very important phenomena, cellular communication. So let's see how cells communicate with each other. So over to Bhavya.
Hello, everyone. So today I'll be talking. Hello. Yeah. Okay. And you can stand. Yeah, not here. Yeah. Yeah. Hi, everyone. So today I'll be talking about how cells talk to each other and how scientists around the world try to study this problem. Okay. So to begin with, all living systems are made up of a basic unit of life called cells. And from trees, plants, to animals, to human beings, we are all made up of these small units called cells. And as you all know, we have the whole body, which is made up of multiple organ systems like the circulatory system, the digestive system, and so on. And within the organ system, you have specific organs like your stomach, and then the tissue. The whole organ is made up of small tissues, which are basically lined up of cells. And similarly, with trees and plants, you have smaller systems, which goes on to small cells, which is the basic unit of life. Now, we all know that we are made up of something called the DNA. As in, DNA is the information carrier for all the living systems, and we know that as human species, our DNA is slightly different from the other species of the animal kingdom. But within our own body, say you take your heart cells or the brain cells, I have a question, which is how so if you look at the cells in the brain, they look like this. These are called neurons, and the cells in your muscle cells look like these. So they all look different. But my question to you is that, does the brain cells and say your muscle cells or your heart cells have the same DNA? Have the same DNA? That is the question. Yes? Okay, so the answer is correct. They do have the same DNA. Then why do they look so different? Why are they so different? Why do your brain cells have a different function compared to your heart cells or your muscle cells? How are they so different if the DNA is the same? Correct. So DNA is like a code, like an instruction manual, which can be read differently by different cells. And it can, so you can have some, say you start from the page 1, some cells start from page 1, some cells start from page 10. And say in a single chapter, if you have a portion cut off, say a paragraph cut out for your exam, and teacher says it's not there for your syllabus, and so you just skip that paragraph. The same way, the cells also can decide to skip some portions of the DNA and transcribe and translate and make proteins of its choice. Now, so as I said, you have different cells, and as a human species also, even though we have similar DNA, we are all different individually. So then how do we make sure that we solve our problems together and evolve as a society? Any answers? Communication, yeah, I mean, is the title of the talk. So talking is absolutely essential for anything to happen for us as well as for cells. And just as another example, you have in a cricket team, you have batters, you have bowlers, you have wicket keepers, and they need to communicate with each other to make sure that the team wins the World Cup. Similarly, your cells in your body need to talk to each other to make sure that you are protected from the external environment. And here is an example. When you touch a safety pin or a needle, a sharp object, you withdraw your hand immediately without thinking. And how does that happen? It happens because the cells in your fingertips talks to the cells all the way to the cells in your brain. And then the brain sends signal. The brain sends the signal back into your hand, your muscles in your hand, which then moves your hand away from the sharp object. And so as you can see, this happens because of cellular communication. And so before I go into how cells communicate, I would like to just briefly introduce what are cells. And from school, you all would remember that cell is like a room which has walls which protects it from the external outside world. Similarly, cells have a boundary called the cell membrane or the plasma membrane. And this plasma membrane is made up of stacks of these molecules called lipids. Now, what the cell membrane is made up of stacks of very interesting molecule called lipids. And what is interesting about these lipids is that it has two parts, a head and a tail. And the head is a portion which loves water, while the tail hates water. So any structure that this lipids form, it tries to make sure that the head is facing the water while the tail is away from the water, protected away from the water. So, so this is the sort of molecules that the cells use to make the barrier of the cell. And if you need a cell to release some signal or send out a signal, it needs to cross that barrier. And so we come to the question, how do cells talk to each other? So for any sort of communication, you have a sender and a receiver. Similarly, the sending cell releases some molecules out into the external environment, which is then recognized by proteins, which is on the surface of another cell. And this protein then binds to these signals and gives the response the cell needs. If the cell does not want to talk to another cell, that cell will not express these proteins called the receptors on its surface, and hence the signaling or that communication will not happen. And there are different types of signaling. In the sense, you can have a long distance signaling that happens from, say, your liver cells to your heart cells, or the cell. So imagine a scenario where you want to talk to a friend who's sitting right next to you, or a friend who's sitting in Delhi. What would you do? Similarly, what cells do is basically it releases molecules into the bloodstream wherever it is, and these molecules, the signaling molecules, then travels throughout the blood in your circulatory system and reaches a cell that is its target, which has the receptors that bind to these signaling molecules. Or if it's it can have also be short distance signaling, where the cells, as you know, cells are all packed against each other, and so there is contact between each other. So if you want, you can have also short distance signaling between cells, which is the same principle, where it releases the signaling molecule right outside, and the cell near it can capture or catch that molecule. Or it can also signal itself, in the sense it can express the receptor that binds to these signaling molecules, and then this can either weaken that signaling event or strengthen it. There's also another type of signaling, which is just contact-dependent signaling, wherein just the proteins found on the surfaces of the cells can bind to each other and signal the neighboring cell. So then the question is, how is this process made? How does the cell find a way to release the signal out into the world? And as I said, the cell has a lipid membrane as a barrier that protects it from the external environment. What it does, it packages the signaling molecules into these spherical balls that we call vesicles. And these spherical balls also contain the same membrane boundary that I spoke about earlier, made up of lipids. And these spherical balls then fuse with the plasma membrane, with the cell membrane, and release the signals outside. And for this process to happen, there are hundreds of proteins, of different kinds of proteins that are involved in making sure that this process of fusion of two membranes happen. So there's one more important step that takes place while this membrane fusion happens for the signal to be released, which is the opening of the fusion pore. As I said earlier, there's a vesicle which has the signal. This vesicle, which has the membrane, this membrane fuses with the plasma membrane or the cell membrane, and there are multiple steps that goes into this process. And finally, what happens is a hole is formed through which this hole connects the inside of the sphere, the ball, to the external environment. And through this hole, you have the signal now getting released into the outside environment. And so scientists are trying very hard to study this fusion pore because it is the door through which your signal gets out into the environment. And if you can control that door, you can control how much of information should be sent out into the world. So the signal can be of different sizes. So one such example of why studying this is important is because the signal can be of different sizes. So you can have a smaller signal to a large signal. And depending on the size of the door, it can differentially release the different sizes, different signals into the external media, external world. And to make this concept a little bit more clearer, I've just used a cat video. So as you can see here, you have a large hole through which the cat easily passes through. But with time, as the hole becomes smaller, the cats find it difficult to move. Smaller the hole, the more time it needs to pass through that hole. So what you can see here is that even though the size of the hole is becoming smaller, with time, the cat is able to push through. So it's a sort of similar concept the cell uses. If it can control the space of that hole, the size of that hole, and if it can control the time at which that hole is open, it can control how much of the signal should be released outside. And when we talk about space and time, we have to realize that this is extremely small. And what I mean by that is the size of the hole can be up to nanometers to micrometers. And when I say nanometer, that is like 1,000 times the diameter of a single strand of your hair. So the size of the fusion pore is about in nanometers, and the time with which the pore is open for is about microseconds, which is 1,000th of a second. And so it is extremely difficult for scientists to study this problem or study how this happens in the cell because it's extremely small as well as it's extremely fast. So, and before I go into how we at TIFR study this problem, I would like to also explain why this is an important problem to study. This is important because this is a process that goes wrong in many disorders, in many brain disorders, for example, autism, epilepsy, Parkinson's, where autism is like when you have lesser signal being passed from one cell to the other, while if you have exaggerated signaling or exaggerated talking between cells, you have problems like epilepsy and Parkinson's. And if we can understand this mechanism of how it happens in the cell, we can probably find a cure for these disorders. Another disorder that can be cured with understanding this process is diabetes. Diabetes is a disease that happens when your cells are not able to release some signal called insulin. And that happens, right. And so if the cells are not able to, and insulin is a signal that makes sure that helps the cells take up glucose, which is in the bloodstream. And diabetes is a disease that happens when there's a lot of sugar in your bloodstream. So if you want the cells to take up that sugar, it needs the command from insulin to say, take the sugar inside the cell. And so release of the signal insulin is important for regulating sugar in your bloodstream. And if you can understand how we can control this release of this signal, we can possibly help cure diabetes. And another disorder that is common and prevalent is asthma. Asthma is a disease that happens in your airways. And what happens there is the immune cells release a signal called histamine. And this histamine is then is a command to the cells in your lungs to make more mucus. And what happens is when, again, the lung cells release mucus, again, which is the process of this membrane fusion and fusion pore and releasing cargo. So it releases this mucus. And what happens is when there's a lot of mucus in your airway, you have a difficulty in breathing. And this is, again, is a problem of how cells, what goes wrong if cells don't communicate properly. So understanding the process of membrane fusion and the fusion pore and how the cells control this fusion pore, the size and the time at which it's open, can help us figure out these disorders. And so here at TIFR, how do we study the problem? What we do is we use current. So we use the help of physics to address this problem, because as I said, in cells, it's difficult to study them. And how do we use physics? So if you remember in school, we have learned that current is basically movement of electrons from one place to another, right? If there's a flow of electrons from one place to another, we call that current, and that is what's current. Similarly, so what we do is we use a system called BLM electrophysiology. The name is not important, but the principle is this. We have a chamber with salt solution. What we do is we create a separation between the chamber, and we create two chambers basically. And this chamber is has a hole in so the dividing wall has a small hole in it. And what we do is we paint membrane on that hole, which this membrane mimics the membrane of the cell. And so what we do is now we plug that hole with the cell membrane. And so now we have separated the two solutions. So now we have two chambers, and in one, so what we can do is we can have different electron concentration or ionic gradient between the two chambers. And only if there's a hole formed now, if you somehow make a hole on that membrane, you can have a flow of electrons from one chamber to the other. And this we can measure as current. And so what we do is on one side of the chamber, we add whatever proteins we want to study that is involved in this membrane fusion. Membrane and proteins, we add whatever material we need to make that membrane fusion or the pore, that fusion pore that I talked about to form. And once that pore forms, so whatever we add some proteins and we make this pore form, we make this pore a hole between the two chambers. And now when the hole forms, what happens is there'll be a flow of electrons from one chamber to the other. And this flow of electrons we measure as current. And this is what our readout looks like. So initially there'll be no current, so you have a zero current. But we add some protein and some membrane, we add some whatever to mimic this membrane fusion, and we get some positive current or some current that comes up. And as you can see with time, that the properties of that pore changes. So for some time it's opening, there's some current, and then it closes and there is no current. Then again it opens for some time and then it's stable. So you can study how the properties of how stable the pore is with time, and the size of the pore. So you can tell from the current, how much of the current is flowing through, you can tell how big that pore is. So such properties of the pore we can study. And this comes to the end of my talk, where what I would like to say is that so studying the membrane fusion and how cells talk to each other is an important problem that biologists face right now. And in TIFR, we use this current system to study in vitro, very minimal system. We are not looking inside the cell, but we're taking the system outside the cell and we're looking at it. And this will have studying this has major implications in figuring out what goes wrong in disorders that I mentioned earlier. Thank you. So if you have any questions, you can ask. And so that is that's more like immune response. So toll receptors are there it's there in all cells, but it's more specific to the ligands or the proteins that it recognizes are proteins from bacteria or virus or some from the external environment, not from within the cell. Within the cell also you have, but majority of them recognize signals from outside our body. Yeah.
So how do the cells know how do the cells know that we have to collect this signal?
Right. So cells have some proteins on its surface which will recognize that signal, specifically that signal. If it has that protein that on its surface, will it recognize that signal?
So for every cell, there is a different protein.
Yeah, not all cells will have different proteins, but if the cell needs to listen to one particular cell, it will have those proteins that it needs to listen to.
Any more questions? If not, let's again thank our speaker.